All right, we'll be doing our project on modeling viral infection. My name's Cam.
I'm Alexis.
And I'm Alex.
All right, so what we're looking at in particular is the herpes simplex virus 1. It's an alpha herpes virus, and it basically can first infect the PNS, which is basically what you would see from like herpes on your face and stuff like that. But it's capable of entering the CNS by crossing the blood-brain barrier. And once it does that, it can establish latency. And when it's in latent state, it cannot infect other cells, but it just sits there, and then it can become reactivated if it's exposed to some certain stress. So yeah, we're just gonna try and model that here. So our question is, how does the virus spread across a given tissue in the brain? And how can we see patterns there? So we'll be looking at both the spread from a CA model and also the largest connected component, which is going to be the connected component is made of just dead cells made from the model.
For our model, we had 3 different states that any neuron could be in. It could be susceptible, infectious, latent, or removed. And for the interactions, we have an infection rate alpha, a clearance rate gamma, a latency rate of lambda, a stress rate of sigma, and a neuronal death rate of delta.
And this is like the model using the ODEs. We're not going to talk about this really, but yeah, it checks out.
And for our model assumptions, we assumed a more neighborhood. Our initial conditions, we assumed that it's a healthy brain and all of the cells and neurons are susceptible to begin with, except for that one, which we assume starts at the top left, but really would probably start on any barrier or somewhere in the middle. And why? Because in theory, when HSV1 enters the brain, it enters via the brainstem, and hypothetically this implies that one virus reaches the healthy neurons first. We assumed a high latency rate, but a low reactivation rate. And the latent state is a special case of the susceptible state, since the immune system can't kill it. And the lattice represents an arbitrary region of the brain.
All right, these are our results. We started with these parameters above, and we see that the virus does move in a branching movement. We did cluster sizes. Now, cluster sizes represent clustering of dead cells. And we see that upon running with these same parameters and initial conditions a few times, we do see variance in cluster size. Another thing that we did was do a change of initial conditions. So we started with, instead of one cell being infected at the top, we did infected cells centered at the middle of the grid. And we do see that the cluster size does increase significantly. Another thing that we did was we changed the parameter, the death rate to 0.3, we increased it, and we do see a considerable change in your cluster size. And some insights that this gave us was that upon changing parameters, we can get different cluster sizes or different sizes of dead cell clusters of dead cells, as well as depending on where the infected cells that start to go from latency into more of an infectious state, we do get different cluster sizes. And depending on cluster size, we do get, or based on cluster sizes, we do get damaged brain regions, which does affect brain functionality.
All right, and then from here, we're not going to really go into depth in all this, but one of the more interesting future directions here would be really evaluating the functionality or the connectomics of the brain when it degrades over time with the viral spread. So hypothetically here, on the far left, you see when the virus is spreading, at each interval of the spread, you could basically percolate some sort of signal across the entire from each axis to another axis and see if it gets basically stopped by like a firewall of these dead cells. And if it does, then you know there's like that it can't basically communicate from A to B, which would be something really interesting to look into. That's definitely out of the scope of this, but then yeah, there's some other parameters you could play around with, like introducing the stress locally instead of just across all the cells. And then you could add more agents like microglia or astrocytes, which just effectively play the immune system. That would definitely cause some spatial changes. And then a larger death rate for like a denser population of infectious, that would just again change like the organization of these clusters. It would make them denser, I would imagine. But yeah, that's it. Hope you guys enjoyed. Thank you.